Thanks for tuning in. This is Scott and Sarah for our first installment of Science Bro, where we bring you the latest in science, the newest in science, sometimes the oldest in science, but nonetheless, we're going science.
Yeah.
And Jack Leg's up in here. What's up? Thanks for joining. Daydreams early. Thanks for coming. We're going to drop some pretty dense science on y'all tonight, so hopefully we pull this off properly. And if not, there's always tomorrow. Here we go. So today we're doing Ecological Monograph, or rather a paper that is found within Ecological Monograph, written by Dr. Elaine Ingham, her husband, and two other people, called Interactions of Bacteria, Fungi, and their Nematode Grazers, Effects on Nutrient Cycling and Plant Growth. And just a reminder that we are doing some live events, and gotta adjust my camera here real quick, bear with me, sorry. And just a reminder, we're doing some regular live events. So our next live stream is gonna be Saturday, January 29th at 12 p.m. Pacific time. We're gonna do part 2 in Sarah's Crescive Method Basic Training Masterclass, which is what we did last Saturday. And on Sunday, again, we're going to do at noon Pacific Standard Time, the Soil Balancing Masterclass, Part 2. And we're going to go over some live examples of mineral balancing and what's in store for our upcoming class. And then next Wednesday, we're going to go to Episode 2 of Science Bro, and we're going to do the same thing we're doing tonight. And so essentially what we're aiming for with this series is to bring people up to speed on how to look up scientific research projects. So when I first met Dr. Elaine in, 2014 or something, an event, I asked her, where can I come into contact with more of your information between now and the next time I can see you? And she said, check out Ecological Monograph 1985. And so when you type in Ecological Monograph 1985, you get roughly 500 results. And it took me a while to even figure out what the heck she was talking about. Eventually what I did was I had a friend that was going through a university upper graduate program in science, and I told him what I was after, and he came back with about 6 publications, and he says, I think you're looking for this one.
It's a pretty big publication.
Pretty big publication, and it's cited, thousands of times by other researchers, so it's a pretty prolific paper. You can see here, this is the website for Ecological Monograph where it pops up. Most research papers or scientific publications will show you the abstract on the first page of what's visible, kind of like if you go to look at a book on Amazon. You can see what volume it was printed in, what year, and what pages it's actually located on. You can download from here, but this is a quite expensive place to do it. If you're a university student, you typically have access to this in your university library, or sometimes, like I used to do, I just go sneak into the library and print it off like I was a student, and just that's what I did. No biggie. But nonetheless, it can be found here, and you go to JSTOR is one place where I've downloaded before. They're a little bit cheaper. You do have to pay for all this stuff. You need a subscription. So in this situation, it says here you can log in through your school library. And those are just a few ways to get it. I'm pretty sure that if I share it, that's against the rules. So we're going to make sure we don't break any rules and go through this paper in a little bit different fashion. I think today we're falling under the category of reviews, I guess. So I think we're within the rules and we can go over this. So
we're just aiming to break down what it takes to break down a scientific paper, basically.
Exactly. Using Dr. Elaine's work, which was insanely effective and important for my personal journey. So first we need to understand the research and the peer-reviewed publication process. So the peer review process aims to subject authors' scholarly work, research, and ideas to the scrutiny of other experts in the same field. So once you've completed a research project, essentially, you submit it for review. Well, first you lock it in under like a trademark thing so nobody can take it, and then you offer it for peer review. And then a board of your colleagues anonymously go through it and essentially tear it apart. So there's the science community itself puts these checks and balances in to make sure that what is being published is of integrity, is accurate, and speaks to the community. Now, with the explosion of the internet and things like that, there's a few other publications that are fairly less scrutinized and a little bit easier to get in, and that's where you get some of the like pay-to-play stuff. But with regards to Dr. Elaine Ingham, when I've talked to her in person on the matter, she speaks to the fact that she has something like 150 peer-reviewed journals, which is unreal. To give you some comparison, one of my friend's dads did some pretty heroic stuff with citrus when we were in junior high in Mexico, and he's an advisor for the USDA. He has 4 peer-reviewed publications, and he literally is a head researcher at University of California, Riverside. He's a big dude, and he has 4. Dr. Elaine has 150, and they're in the top 2 or 3 publications in the entire world. So the goal of the peer review process is to encourage authors to meet very high standards, to minimize erroneous claims, answer meaningful questions in the field, things like that.
Continue research that's already been going on.
Exactly.
Yeah.
And there's 2 main goals of this process. One is to act as a filter to ensure only the highest quality research is published, determining validity, significance, and originality of the study itself. The second goal is to improve the quality of the manuscripts that are deemed suitable for publication. So sometimes they request a revision or ask for further clarification. So kind of like when you were in school and you were putting together a science project and you would run it by your professor or your teacher, then they would sometimes suggest a few changes here or there. Once you go through this back and forth with the board, then you can make a published peer-reviewed journal.
Yeah.
And for the most part, every scholarly article or peer-reviewed journal will contain the same basic components.
Yep.
On the first page, they have the abstract, which is essentially a summary of what's going on, which is what was shown on the JSTOR website and the Ecological Monograph website that shows the first page. It's so that as you're flipping through the journals as a researcher or a scientist or a citizen scientist, as we like to be called, you're looking through the basics of that to see if it applies to you and you want to delve into it further. Then there's the introduction, which explains why you're doing this in the first place. What is it that spurred the concepts? What other research is prompting you to explore further? Which kind of ties into the third part. When you produce your peer-reviewed journal, you also have to include other works. So you do the abstract, you do the introduction of your goal, and then you start to cite other works that are similar or maybe less complete than you would like to see, and you're simply expanding on what was there before you.
Kind of like building the case. So-and-so did this much research that posed a question to us. We're going to run with that and expand it a little further.
Exactly.
Yeah.
And then part 4 is the materials and methods, which is exactly how you did the research paper, which is normally where I jump to. So I read the abstract and then I jump to the materials and methods to see how much of the scientific process was actually applied. And that sounds kind of crazy to say, that you'd think that a peer-reviewed journal would be of the highest integrity in science, but sometimes even me, I'm able to read that there's some information intentionally left out.
Or limitations in the way that they came
exactly.
to the conclusions.
Which is actually more common with refer to biology. So for in our case, when we read publications that are in regards to soil biology, most often they're using the plate count method, which from my viewpoint is fairly worthless unless you're examining pathogens. So I go to the materials and methods first to see what is it they did. Then they present all the results, which is usually lots of graphs, data, charts, things like that, what happened. And then they discuss the results. So what does it mean to them? What do they think it means to the field of study? Things like that. And then in the conclusion, they just extrapolate on what was learned, further extending. And sometimes those results, discussion, and conclusion don't necessarily have a title or a header. They're just kind of blended in towards the end in that general order. So today we're going over this one, which is Ecological Monograph. which when Dr. Elaine first said this to me, of course, I was smoking a little reefer at the event, and she said, look at Ecological Monograph 1985. I'm pretty sure I wrote down Ecological Monograph 1986. I have a heck of a time finding this, and I was a long way from this whole shenanigans. So this is the paper that we're talking about today. And for me particularly, I've always been the nerdy cannabis guy, where within my crew or my peer group, when something wasn't understood, I was the one that would try to go find the answer from a scientific basis. So I feel like for somebody that's into cultivating cannabis, I'm fairly suited to doing scientific research. And when I got to this one, there was some stuff that I just had no idea what the heck they were talking about. So this particular paper, for example, is 22 pages long. Most peer-reviewed papers are 3 pages long. So it was quite dense, quite long, quite complicated. So it actually took me about 2 weeks of going through and highlighting different definitions, trying to figure out what the heck it meant in the context of the paper. And honestly, it didn't really make complete and total sense until I actually got the chance to sit down and talk with Dr. Elaine and ask very specific questions, of which I'm insanely grateful for. That was some of the most amazing moments of my adult life, being able to sit down at the dinner table at the ECI farm with James Carroll and Dr. Elaine and throw back a couple porters and talk nerdy microbe stuff.
And me.
And Sarah. Yep, Sarah was there too. That's where we actually met. But so there's some concepts that are really hard to wrap your mind around because the terms seem kind of opposite of what they're talking about. So in the case of mineralized, they're talking about the conversion or breaking down of organic compounds, meaning it has a carbon attached to it, into smaller inorganic compounds, meaning it doesn't have a carbon attached to it. So breaking it down into its simple nitrogen forms, something like that. So mineralized, mineralized would be the first step of taking a rock and turning it into plant food. Remineralized, on the other hand, is the conversion and breakdown of the organic nutrients, so same concept, starting with a carbon-based element, breaking it down into its inorganic form, but back through again through what we affectionately refer to as the poop loop.
Yes. So the bacteria will bring it back into their bodies, and they'll get eaten again, and it'll remineralize, and then that can keep going and keep going and keep going.
Which is how these nutrients are produced for plants in the soil food web system. So you have a protozoa that has a carbon to nitrogen ratio, and it eats a bacteria that has a narrower carbon to nitrogen ratio. So in the effort to get the number of carbons it needs to reproduce its body, there's excess nitrogen. It's almost like when you're doing division in elementary school and there's a remainder. That remainder are those remineralized nutrients that can go back into solution and become available for the plants. So those are kind of confusing, but those two terms are used a lot. So those are beneficial things. Mineralized means making it into plant food. Remineralized means coming out the back of another organism. Then we have immobilized, which I thought was a bad thing. And this was the one that was really confusing to me because immobilizing is the conversion of organic matter into organic matter, same thing, but then held in the body of an organism.
Organic matter into inorganic. Yeah. So this is what I was talking about. If anybody tuned in to the last session where somebody had asked a question about feeding alfalfa to their as a mulch, basically, and that you would have a bacterial bloom. And I was saying, well, you can tie up all your nutrients because they'll cause a bloom of bacteria. And if you don't have the predators in the system to mobilize the nutrients in that poop loop to remineralize, then you can immobilize all those nutrients into the bodies of the organism, into the bodies of the bacteria specifically.
It's not as extreme as what hydroponic people would define as locked out, but they are fairly unavailable and jammed up. So that's why we don't rely on just bacteria. Another concept that's kind of interesting is called the p-value, which is a measure of probability and statistical significance, or the variation from what we would call like the standard deviation, which is kind of like an average of the data set. So if you had 20 samples that were between 80 and 100, and you had one outlier of 35, that would be a high p-value. So the p-value is like a percentage. So if it was 0.50, that would be a 50% probability. And to be considered legitimate for peer review type publications, you really got to see the data at a 0.05 or better, which would be 5%. Pretty much everything I think in this paper is of 1% or less.
Which is
insane.
Really insanely accurate and reliable data.
Which this one part is what drives me nuts about people objecting to Dr. Elaine. It's like, this was in 1985, y'all. Like in 1985, she understood this to a measure of probability of less than 1% error. And we're in 2022 still wondering if she knows what she's talking about. Get out of here. So the abstract is extremely large, and I didn't want to put the entire thing on and read you 1,000 words, but here are a few sentences out of the abstract that is what originally grabbed my attention and what we think is most important to pull from that.
Yeah. This was pretty fascinating. All treatments containing nematodes and bacteria had higher bacterial densities than similar treatments without nematodes. She had mentioned this to us when we were up at the first consultant training where we all got together, where the nematodes can actually, basically, when they eat some of these bacteria, they're not all absorbed, and they tend to stimulate and come out the back end and stimulate the populations of the bacteria, kind of like stimulating their food source. So maybe it's a, survival of the fittest kind of thing, and just, I don't know, a mutualistic thing going on. But anyway, this is a phenomenon that we actually see where nematodes can actually maintain the populations of the bacteria fairly well.
It made me not fear death, I'll tell you that. I honestly, that's when I stopped worrying about dying and reincarnation. Because that's pretty...
Bacteria inspire you?
Yeah, it really did. Yeah. Like I could watch while I'm sitting there, things get eaten, look like they're dying but not dying, and keep on moving and actually improve things. So what Dr. Elaine said about the nematode phenomenon was the nematodes are larger and they can move a lot farther than a bacteria could ever dream of in its life cycle. So they're actually eating bacteria and typically moving towards an area with a higher resource of food. So the bacteria gets eaten out on the edges and actually gets dropped off in a better place to hang out. So wrap your mind around that one.
Yeah. All right, so this is another part of the abstract. Plants growing in soil with bacteria and bacterial-feeding nematodes grew faster and initially took up more nitrogen than plants in soil with only bacteria because of increased nitrogen mineralization of bacteria, ammonium excretion by nematodes, and greater initial exploitation of soil by plant roots.
So essentially what she's saying is more nematodes, more roots, more fruits, more yield.
Yeah.
And
so the plants, having the bacteria with the bacterial feeding nematodes, that specific grazer, actually bloomed more bacteria, released more nitrogen into the soil, and the plants were able to take up more nitrogens. It stimulated the whole system, the entire microbial ecosystem, as well as the plant interaction with that. So there's something going on there that's much more than you can't just reduce it into the parts and think that when you add it together, 1 + 1 + 1 = 3. This is more like a 1 + 1 + 1 = 5 kind of effect. So
exactly.
That's kind of what they're saying here.
Nematodes growing bacteria, making their own food source, which is pretty crazy.
Yeah.
This was kind of trippy.
This was very trippy. So it also said the addition of fungal feeding nematodes did not increase plant growth or nitrogen uptake because these nematodes excreted less ammonium than did the bacterial feeding nematode populations, and because the nitrogen mineralized by the fungus alone was sufficient for plant growth.
Which I guess we should define here. So the two predominant forms of nitrogen are nitrate and ammonium. The solution hydroponic-based concepts and mentality of the university system focuses on nitrate, and they actually kind of poo-poo the ammoniacal nitrate forms. And this is where I go all in on Team Dr. Elaine, because under a fungal dominant system, like a true beneficial fungal dominant system, the form of nitrogen is actually ammoniacal nitrogen, this NH4 plus -N shenanigans. over nitrate, and it has to do with the acidity and the alkalinity of the soil and what's being produced in that. We thought about including a slide on that, but I feel like that could be a whole 30-minute conversation, so we'll come back to that.
Yeah.
But nonetheless, when it's bacterial dominant or anaerobic, there tends to be nitrate dominance, and when it's aerobic or also typically fungal dominant, you get more ammoniacal nitrogen. If you look on the back of any grow and bloom hydroponic formulas, you're going to see more nitrate in the veg and more typically ammoniacal nitrogen in the bloom. If you tried to run the grow formula from start to finish in hydroponics, you'd have a buggy mess. A lot of people figure out that you can run the bloom formula from start to finish and still have decent results. So this is why that is. You can run the ammoniacal nitrogen earlier, but you typically can't run the nitrate nitrogen into flower without major issues.
Yeah. All right.
Yeah.
There we go.
I was like, wait a second. I lost myself in my show. We'll get this dialed.
Okay. So these were the objectives stated in the study. 1, to propose and evaluate a conceptual model in which the levels of nitrogen and phosphorus in microfloral grazer populations are considered as separate state variables in the nutrient cycling process, as suggested by evidence in the literature. And then 2, to examine the interactions between bacteria or fungi and the nematodes that feed upon them and how these relationships influence nutrient availability and plant growth. So that's the objective, pretty narrow on this particular study, but there was a ton of information in this study that we're about to go into. Where the who's who, which I think is the next slide, right?
Yeah, so essentially what they're saying is they want to find out with different combinations of soil organisms what happens to nitrogen and phosphorus and what happens when nematodes and fungi interplay with each other.
So they're tracking that through the system.
And this is what really grabbed my attention, because at this stage in my development back in, I don't know, 2013, 2014, whenever that was, you know, most cannabis is looking to get phosphorus. There was always conversations about bloom boosters and hardeners and phosphorus. And this was where I really got locked in that, like, this Dr. Elaine lady was onto something, and it can significantly benefit cannabis, or really all plants, but at that, obviously
That's what you were interested in.
That's what I was in, yo.
Yeah.
Yeah.
All right, so the current study examines the interactions of the biota represented in the model in order to substantiate previous observations under more rigorously controlled conditions. So here we are talking about previous work, now they're going to take it a little further. And with a more complex assemblage of organisms, including plants. Nitrogen and phosphorus mineralization were examined in 2 model soil systems, one in which soil was unamended and one in which chitin was used as a representative organic nitrogen substrate. Bacteria, fungi, and nematode grazers of these microflora were chosen from isolates to represent functional groups operating in natural soil systems, and they used the blue grama grass for this.
Right. So they took soil, they sterilized it, put it into a petri dish. I believe one of the other slides talks about this, but replicated it 5 times. So they had 5 times of every single combination of organisms, and then they inoculated individually. Their goal was to do bacteria, fungi, and nematodes, but in this process, protozoa snuck in. And that was something Dr. Elaine told me around the campfire, so to speak, was that they were really trying to do just bacteria, fungi, and nematodes, and in the process, some amoebae and whatnot snuck in there. So you have to include that and in the data and whatnot. But Dr. Elaine's husband, who's also on the paper, is a nematologist and a research scientist. So that's where that was coming from. And then they also added chitin to see how that affects. So people do insect frass or chitinase. You might be familiar with those. And that's essentially what we're doing.
Yeah. All right.
Coming in hot. We'll get there.
All right. So this was a little excerpt from where she's mentioning other work being done that they're building upon. And like I had had mentioned a little bit earlier, this is that little excerpt that literally states, bacteria can also act as a nutrient sink in soils. With increasing additions of glucose, more inorganic phosphorus was immobilized by bacteria, and at the end of the 160 hour, there was no net mineralization of phosphorus. And then this last sentence is really the clincher here. This guy Coleman, in 1977, found that almost all soil ammoniacal nitrogen and 40% of the initial phosphorus were immobilized 21 hours after inoculation of bacteria into glucose-amended sterile soils. And then it goes on to talk about, what happens after that. But I just wanted to point out here that when I say that you can tie up the nitrogen and nutrients in your soil if you don't have the predators in the system, and when Dr. Elaine says that, that's where that's coming from. Work done previous. And so here it is.
This is well known. This is before her. This is 1977. And what that means to the cannabis farmer is that in the community, if you see, somebody that relies on a bacterial product and is heavily influenced by the saturated paste test, they want to see that soluble phosphorus like 2 to 3 parts per million or more. That's because 40% of it is tied up in the EM1 and tied up in the bacteria not being released. In our system that we do with full predator-prey interactions and very high numbers, you can get better results or equal results with like 0.5 part per million to 1.5 part per million phosphorus.
Yeah, and it's also like a lot of these broad acre, like I know maybe not everybody's doing broad acres, but if you're doing broad acres and you're only adding in these bacterial inoculants and mycospores, or you're only adding in manure, manure is pretty much only bacteria in the manure. And so what you're getting is bacteria, and you start to wonder why these soil mineral tests aren't proving to be reliable. Well, if you're not working with a soil food web and you're only using the Haney test to try to see whether or not you've got, your microbes in there, you might be missing a few of the microbes. And in my experience, in our experience, when we're looking at a lot of these tilled big acre agricultural fields, they're almost all bacteria and very, very, very little, if any, predators in the system. So just something to consider.
And a great point by Smiley's Garden. This is what people seem to skip over. The biology needs nutrients too. It's a system, and they're all functioning. So.
Yeah, it's how they generate energy to do what they need to do, their function in nature. So, all right.
So this is the second part of it, just below.
Okay, so we moved on here. So when either the amoeba or the bacteriophagic nematodes were introduced to some of the microcosms, nearly all of the immobilized nitrogen was remineralized, while less than 1/3 of the ammoniacal nitrogen was returned in the treatment with bacteria alone.
Let's stop there for a second.
Okay.
So bacteriophagic nematodes means bacterial feeding.
Yes.
So when you have either an amoeba, which is a predator of a bacteria, or a bacterial feeding nematode, nearly all of the nitrogen that they measured at periodic dates through the 40 days was remineralized, meaning went through the poop loop and was then reavailable.
Yes.
In the situations where there was just bacteria, less than 1/3 of that was put back into solution and available to plant roots.
Yeah.
This is the part that that's when I knew I was onto something, and I knew that there was something completely missing in the cannabis industry. And at that point, there was Rocky Mountain Worm Company, which was actually making a biologically complete compost. Everything else at that point was bacteria and maybe an amoeba, maybe a piece of fungi, pretty much just bacterial compost. And I was like, where the heck do I find nematodes that eat bacteria? And it took me a couple years before I figured out, it took till 2016 when we started getting into the big commercial projects where they were introducing other predatory nematodes, and that's where we got turned on to HB nematodes. So I first read this paragraph in 2014-ish and was like, damn, I gotta get me some bacterial feeding nematodes. Come to find out, the predator insects people were already handing them out. You just had to get the right one. Yeah. So most people are after the SF or the SC. We went with the HB nematodes, and that made all the change in the world. Yeah. So if you are a person that's doing EM1 only and saturated paste tests, y'all need to get the right nematodes. And yeah, this is live. We're going right now.
Yep,
we got some other live stuff coming up. We're going to try and do this every Wednesday.
Yeah.
So tune in.
Yeah, Dr. Elaine had told us back then in that soil, her first consultant meeting, that HB nematodes were very important in the system for very specific reasons. So anyway, so we'll digress and go back to, or not digress, and we'll go back to the
so after
slide
24
here.
days.
Okay, so after 24 days, significantly more phosphorus was remineralized in the bacteria and nematode treatment than in either the treatment with bacteria alone or the bacteria and amoeba treatment. In a similar study, more nitrogen was remineralized as ammoniacal nitrogen in an amoeba plus bacteria treatment than in a treatment containing bacteria alone. A treatment with nematodes and bacteria, or with nematodes, amoeba, and bacteria.
So again, referring to previous research as old as 1982, that was 40 dang years ago.
Yeah.
Still wondering if we need to measure bacteria.
And all the rest of the soil food web.
Come on,
need
fam.
to do that.
You gotta get with it. 2022 is the year.
All right. Okay, so this is just talking about the design setup, so we can see how they did it. So I'm just going to read it. The design for the unamended experiment consisted of 6 treatments. So 1, a sterile plant control, which is P, plant and bacteria, which is PB, plant bacteria and bacterial feeding nematodes, so PB and B, plant and fungus, PF, plant, fungus, and fungal feeding nematodes is what that is, mycophages. So that's PF and F. And then plant, bacteria, fungus, and bacterial feeding nematodes and fungal feeding nematodes. So that's P, B, F, NB, and F.
The whole party.
Yep, the whole party. Each treatment was replicated 5 times. So now we have replication in the study and sampled once after 40 days.
5
so data,
times.
5 times. Data collected included shoot and root biomass, soil bacterial and nematode numbers, and soil ammoniacal nitrogen and bicarbonate extractable phosphorus. Excuse me.
I don't mean to be obnoxious, but in 1985, Dr. Elaine and her husband replicated this phenomenon 5 times, y'all.
Yeah, they did.
It's
And
2022.
You got to in order to
It's
get.
2022, y'all.
Yeah.
Okay.
Okay.
So just to reiterate, and a little bit easier to understand, here's the different, they call it assemblages. So on our next graph, we're going to use these acronyms. So the P is the sterile control, and then all the other different assemblages. So this was a graph that I've used in some of my presentations that was the really impactful component for me, because I saw the cannabis life cycle in here. So this first line is black. This is the above ground biomass of blue grama grass grown in a petri dish with just the plant and sterile soil. And at periodic intervals throughout the 100 days, I guess it would be, they would harvest some of the replicate samples and measure the above ground biomass. They would measure the underground root zone, weight, and all these things. For this focus, we're just going to talk about the above ground biomass. So when we add plant plus bacteria, you do get a significant increase. And when I was on my living soil journey, moving from, oh damn, sorry, I had the music on the whole time. My bad. Rookie shit. Sorry about that. Thanks, Daydream. Last time Jared recommended me, that was the one.
That is live. Now you know.
It is what it is. We're live, y'all. Sorry about the music. But anyways, this is plant plus bacteria. And so when I was on my living soil journey, first thing you run into, EM1, it's a gateway drug, and you put it in and there's a considerable improvement. Well, here you go. It does provide considerable improvement. When you add fungi, there's a massive jump. And you can see here that I added the green line here. And so this green line is what I loosely interpret the vegetative cycle of cannabis, roughly 30 days, 3 to 4 weeks. And what I saw was right here, most people are in a sterile soil, might have some bacteria. All I got to do is get some fungi in there and I can damn near double my above ground biomass at this 30-day marker, or what would be the end of turning a clone into an 18-inch tall teen. And that was really influential. That was huge for me. I saw that and was like, okay, we got to figure out how to do this. Because in this moment, I saw the issue with why organics was typically not known for keeping up with hydroponic growth rates. And so I thought we need to figure this out and figure out how to get this fungi in there, and so then we can catch up to the hydroponic rates. When you add bacterial feeding nematodes, you get another jump, again, fairly similar to the plant with fungi. The plant with the bacterial feeding nematodes are increasing, I'm glad you're enjoying the music. I think it might be distracting. We'll find a happy medium. We'll probably do some music ones and we'll do some non-music ones, but I'm glad you were enjoying it. Thank you. So,
all right, so back to
with adding the fungi, it's live, come on, man, we'll get it right. We're just going to keep improving, man.
Yeah.
You just show up and it is what it is. So when adding fungi, you get aeration to the root zone. Aeration to the root zone increases flowering nitrogen and above ground biomass. When you add bacterial feeding nematodes, you're now cycling nitrogen and cycling phosphorus,
yes.
which is huge, especially if you're talking about cannabis. So now, if you add fungal feeding nematodes, not much change. And one of the things that really hung up on me, because I'm a nerdy guy, I am into scientific studies, and what I noticed was all this effort, and at the end of the 100-day study, there's a very narrow gap in the above ground biomass between plant plus bacteria and this blue one here. This is the whole assemblage. And that was really messing with me. And when I got to sit down with Dr. Elaine, I had to ask her this. I was like, from my analysis of a graph, this isn't that great of a selling point. What gives? And she says, Scott, remember, we're growing grass in a petri dish.
Can only grow it for so long.
Where can it go?
Yeah.
And so another reason why I put this green line on here is the 80 days is generally accepted full harvest cycle period for cannabis, from veg to bloom, 8-10 weeks, something like that. And if we allow this to be a proper root zone that can continue to expand, we can see that from here in a normal growing conditions and not growing grass in a petri dish, it's probably going to be significant. And in our work, 100% of the time, this is the case. And, we entered the commercial cannabis market in 2016, and we were working behind other consultants. We were working behind other brands. And at that stage of our development, it was really better for everybody. We didn't have to worry about being forward-facing. We didn't have to market our business. We just had to show up, help other people that were in the space execute, and make everything better and continue to improve our skills. And that's exactly what we did. And in 100% of the situations from 2016 to 2018, simply adding biologically complete compost or biocomplete compost or inoculum grade compost, whatever you want to term it, compost that actually has the proper combination and quantity of organisms, when we added that to a commercial farm, off the top, first harvest was 30 to 40% increase in yield.
Yep.
The only time we had a decrease in yield by adding this was when the farm didn't believe us that there was going to be double the biomass around week 3 to 4. And 100% of the time that they didn't believe us, the plants actually grew into the lights and actually caused a lot of problems, and that was the only time that there was a decrease in yield. In 100% of those situations, the farmer was like, I'm not even mad. We just grew the plants in the damn lights. We just got to adjust the schedule. And from there forward, people started to believe us that you could reduce a week of veg. And I mean, that was with the literal top in the industry that most people reference as the influencers and bigwigs in this living soil game. Mm-hmm. And when Sarah came in and did her analysis and added our compost, right off the top, first harvest cycle, 30 to 40% increase. And that's what this is all about. When we look at the cannabis community at large from a numerical standpoint, and we think of it from like an F to B ratio or a basic number, mathematically, most of the industry is about halfway between this black line and the green line on a organism population. And so all we got to do is get y'all up here somewhere. And I would say in 2022, we're like up here now. We're really starting to get the hang of this. people are, there's more availability of, everything's just getting better, we'll
I
say.
would just say that the world, almost everybody is at the green line where it's just bacteria.
Yeah.
And you can do bacteria and mycospores, but that's not really going to help you that much. So, it's not, I'm not saying don't use them. I'm just saying, look what you're leaving on the table, all that.
Exactly.
On the table, so.
It's not bro science, it's science, bro. And here we go. So,
All right.
say what's up to everybody in the chat. Got to make sure everybody's included.
All right. Okay, so back to the study. Shoot and root biomass. So shoot production was, one, significantly higher in treatments with bacterial-feeding nematodes or with fungus than in the sterile plant control or plant and bacteria treatment. In addition, the most biologically complex treatment of the PB, F,
The whole
NB,
shebang.
and F had more shoot biomass than all other treatments. Root production was significantly higher in the plant and fungal treatment than in any other treatment.
So this is kind of an interesting one
Yeah.
because at that bottom sentence, root production was significantly higher in the plant with fungal treatment than any other treatment, which is kind of interesting. I think
Yeah.
that has to do with oxygenation. If you think about hydroponics, you can pick up the bucket and look at the roots. You can't do that in living soil. And when I read this part, I figured out this is how we determine root health and oxygen in the root zone when we're doing living soil. Another thing that's really interesting is shoot production, which is above ground biomass, was significantly higher in treatments with bacterial feeding nematodes. I'm telling y'all, this is super easy. You just got to get some HB nematodes. Don't excessively apply them because you will now cause a boom bust situation, which we're trying to bring onto the mindscape of the community, that if you add too much of one organism, you'll actually collapse it like a house of cards.
Yeah. There's going to be a threshold there. We don't know where that threshold quite is, but we've got an idea.
Pretty much a safe application rate is around what Sound Horticulture recommends, about 1 million per 1,000 square feet. If you stay that or below, you're pretty safe, but I would not go over that. Which kind of sucks for the small gardener because a lot of times you buy $40 worth of nematodes and I tell you to use like a third of it. People aren't usually hip to that, but that's really what you got to do. Maybe someday we'll get Sound Horticulture to offer like a micro batch. So there's a question here which is kind of interesting. What about mixing all three species together? What do you think about that?
All three, the ones from Sound Horticulture?
Yeah, SF,
Yeah.
SC, and HB.
Sure. That's not a problem.
Typically, like we used to order them from Amazon, come in powder. There's other companies that blend them, and we found that getting the right ones and getting live ones and getting ratios were the most problematic when they were blended.
Yeah.
We don't see that with Sound Horticulture. They're doing a really good job with their technique, so they're viable and alive. What happens when you blend them is essentially they're all after the same food resources, so some of them start to fall off. So we typically go with the SF and the HBs, and over time you would see some of those drop off, and then you typically re-inoculate with just one. That's typically what we see.
Yeah. Okay, so both treatments that included nematodes had significantly higher bacterial numbers than the treatment with only plants and bacteria. And the highest bacterial densities were in the plant bacteria and bacterial feeding nematode treatment. Soil inorganic nutrients, there was no significant difference in soil phosphorus between any pair of treatments. All treatments with bacterial feeding nematodes or fungi contain significantly more ammoniacal nitrogen than the sterile plant control or plant and bacteria treatments.
So what that means, again, that's saying that somehow the bacterial feeding nematodes increase the population of their food source, which is kind of trippy as long as you don't over apply. And then again,
I don't think they're doing them in the millions here.
Yeah. We're getting, and again, we're getting more flowering nitrogen, more above ground biomass when we put bacterial feeding nematodes or fungi into the sample.
Yeah, that's a case where context is important.
Context is the most important. Most important.
All right, so after day 21, root biomass in all treatments with chitin decomposers was always greater than in the treatment without microflora. Among treatments with microflora, root biomass was higher in the plant and bacteria treatment than in the plant, bacteria, and fungi, or plant, bacteria, fungi, and fungal-feeding nematode treatments on day 77, although it was not so on day 49 and 105.
So this one's kind of complicated to
Yeah.
digest, and this was one that I made a lot of sense after I got to talk to Dr. Elaine with it. And essentially there was an ebb and flow of root biomass from day 1 to day 105, which was just kind of fascinating. Essentially what it's saying is adding the chitin decomposers, so that would be fungal feeding nematodes, or I would believe an amoeba or protozoa would be a chitin decomposer. You get more root biomass. So in that context, it's meaning that the availability of nutrients and the decomposition of chitin, so insect frass, chitin is what makes up the fungal hyphae, leads to expanded roots. I think most people are familiar with insect frass providing benefits. Where it gets confusing is on the plant with bacteria, it had the highest root biomass on day 77, so 2/3 of the way. And I asked her, I was like, how is that beneficial? And she said, it's not beneficial. It means the plants were searching further for nutrients. So in this situation, the large root biomass in the plant with just bacteria was an indication that the plant was taking the above ground biomass energy, or rather the energy that would grow above ground biomass, and spending it on root zone.
Searching.
So in a situation where you're relying strictly on bacteria, your plant is spending energy building roots, not building fruits. Y'all.
Okay, so the main effect of grazing was significant with a P of the less than 0.01. All plant treatments immobilized phosphorus, with the least immobilization in the sterile soil and the soil with bacteria treatments, and the most rapid initial immobilization in the sterile soil, bacteria, and bacterial feeding nematodes. By the end of the experiment, however, the phosphorus values were similar for the soil plus bacterial feeding nematode, soil, bacteria, and fungi, and then plant, bacteria, fungi, and fungal feeding nematode treatments.
That's a lot to digest.
Yeah.
So essentially what it's saying is, so tying up in the body happened the least in the samples with the least organisms, plain and simple. The fastest positive tying up, so tying up in a positive fashion of nutrients in organism bodies, happened in the bacterial feeding nematodes.
Yeah, with the least immobilization in the P. So, yeah.
Yeah.
Okay.
So again, get the nematodes in, get them in veg.
Yeah. Ammonium nitrogen increased slightly between days 9 and 21 in the uninoculated control, and then remained constant for the rest of the experiment. Both the bacterial feeding bacteria and fungi and the bacteria, fungi, and fungal feeding nematode treatments showed significant net mineralization of ammoniacal nitrogen, with no difference between the two treatments. Over time, all plant treatments immobilized significant amounts of ammoniacal nitrogen, although there was some early net mineralization in the plant plus bacteria and plant, bacteria, and fungal treatments. The most rapid immobilization occurred in the 2 grazed treatments.
So in this situation, when they're talking about net mineralization, they're talking about a positive increase in the availability of nutrients from organism to organism interactions. So in the uninoculated control, there was a slight increase of ammoniacal nitrogen from just basic nitrogen reduction, and then it stayed flat, so no more was being created. Pretty straightforward there. Significant increase when there's fungi or fungal feeding nematodes, because fungi are the ones that produce that ammoniacal flowering nitrogen, and clearly the thing that eats it is releasing it. So yeah, and then again, the most rapid reaction of this process happened in the more complex. So again, if all you got is bacteria and you add a fungal or a bacterial feeding nematode, you're damn near matching bacteria, fungi, and fungal feeding nematodes, which is actually a really hard organism combination to attain. But it's super easy to throw a nematode that eats bacteria on top of bacteria you already got.
Yeah. The most significant increases in available ammoniacal nitrogen Plant growth and plant nitrogen occurred when bacterial-feeding nematodes or fungi were present in the unamended experiment.
But that's So again, we made this giant so that you don't get it twisted. The most significant increases in flowering nitrogen, above ground biomass, and plant available nitrogen occurred when a bacteria feeding nematode was eating a bacteria or beneficial fungi were present. And I think maybe this is a good time to clarify what she means by the word fungi here.
Oh, yeah. Well, she's certainly talking about fungi that are non-pathogenic, that are beneficial to the plant itself. So we're not talking about any pathogens here.
Mm-hmm. Here's an interesting question that might apply is, could you keep up with nematode boom with molasses for the bacteria? So the question is, can you throw a food source in for the bacteria to keep up with the bloom?
Wow.
You could do that, but it'd be very complicated and...
Okay, so I have some thoughts on this. So molasses is very stimulating to bacterial populations, incredibly stimulating to bacteria. So I think you could try to do this, but I don't know. I mean, what do you think?
Well, it's like saying, could you start your car with starting fluid? It's like, yeah, your car will start and it'll start aggressively, but it could be very problematic.
Or starting with jet fuel. I mean, like
So jet fuel.
Yeah.
What I would do if you wanted to combine these ingredients together and use this as a strategy, what I would do is I would conservatively apply the bacterial feeding nematodes and very conservatively apply the molasses after and leave it there. And when I say conservative, that's far less than most people are used to. So like we've had fantastic results stimulating bacteria populations with very small amounts of molasses.
And here's the other thing to think about. So nematodes reproduce every 2 weeks. Bacteria reproduce every 20 minutes. So you're not going to be able to chase down the bacteria for a very long time, and you will have tied up a whole lot. You will have immobilized a whole lot of nutrients in the system until you get to those populations, unless you want to try. I mean, I don't know. It's not really the way that we like to go about it, just because we're a little bit more controlled about the way that we do it. But it's just something to think about when you're playing with these different populations.
This is an interesting question about adding protozoa to the mix. Which, that's absolutely. That's one of the things that we're really after with our compost, which they are present in there.
Yeah.
As far as other commercially produced bagged composts, pretty hit and miss. There's only a couple brands we've seen that tend to get the actual protozoa in there. At the large ag scale, you can buy a 275-gallon tote with bacteria and protozoa in it, which is kind of crazy. Yeah. I don't know that that's available on the small scale. Somebody should jump on that. Maybe somebody like Sound Horticulture could produce that. We got our hands full trying to keep the music off during the live stream. I don't know if I want to be another type of microbe herder, I guess. We're already kind of...
There's one more thing I wanted to say about that, adding molasses in and then trying to wrangle in those bacteria with the nematodes. The thing you also have to consider when you bloom bacteria like that with molasses is that when they reproduce every 20 minutes with that stimulating food, they're using up oxygen and they're going to use it all up, and they're going to keep reproducing, and then you're going to go anaerobic, and you'll swing who shows up to the table. So it's playing with fire, in my opinion, when you use molasses. We use it very sparingly, and when we do, in very low doses, very low doses. So
typically,
anyway.
I mean, most people seem to want to use like ounce per gallon type thing, so that'd be 240 mils per gallon. We've had fantastic results with like 1 mil per gallon, 2 mils per gallon, and like not very often. Yeah. So
we find it's typically not totally needed.
Yeah, and this is how we used to do it. You can do it with hay. Hay's a little difficult because it's kind of hit and miss, so you really got to be on your game and watch it and be willing to spend 2 to 4 days dinking around with it. Um, you know, we're definitely not at all advocates of neem, but one of the easiest ways to make a protozoan infusion is with neem cake, but you can also make a tremendous problem. But on any high nitrogen material, there's going to be bacteria and protozoa or the predators of bacteria. So cakes, nitrogen cakes, so neem cake, which again is antimicrobial and can be problematic. It's a very paradoxical product. But, wet grass with dew, you can do it, which is kind of difficult. But essentially, here's the concept. Less is more with bacteria because you got to understand, nematodes, I think, are like some of the slowest growing animals in there, and they're like a 2-week time period. Mm-hmm. When we do a microscope assessment that takes 45 minutes, we've seen 2.5 life cycles. You're talking about a 12-hour life cycle or 12-hour light cycle each day you're growing plants. There's a tremendous amount of 20 minutes in there. Even if it's an hour or two, you're talking about many, 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 many life cycles.
So you can see there's a lot of variables to keep in mind when you're trying to really just keep things in balance without going crazy in any one thing. So
this is interesting. Can the increase in nematodes then decimate the protozoa population? And indirectly, they do, because if they collapse the bacteria, which is the food for the protozoa, we also see, like the one you did yesterday, saw that.
Well, if you throw in 1 million when there are only, when there aren't enough bacteria to necessarily even out, so now we're talking grandiose amounts of nematodes, then I suppose you could overgraze and graze out their food source. But, it's just all context. Whatever you're talking about is the context. So there's, we are talking about predator to prey interactions. So, just depends on what exactly those numbers are.
Mm-hmm. So this is kind of like the conclusion. So this is towards the end and just kind of a summation of what happened.
Okay, so in the short term, I'm just going to read it over here. In the short term, unamended study, there was net nitrogen immobilization in the plant and plant plus bacteria treatments as the plants took up the limited available nitrogen while little or no nitrogen was mineralized. In the treatments with fungi and or bacterial feeding nematodes, however, there was more nitrogen mineralized than the plants could take up, even though there was greater and more rapid plant growth in these more biologically complex treatments.
So let's break that down real quick because it's deep. So essentially what they're saying here is in the sterile and bacteria-only treatments, the nitrogen was coming available much slower than basic grass seeds in a petri dish were consuming. So there was no creation of nitrogen. And I think this is one of the reasons why people start to have resistance towards what Dr. Elaine is talking about. If all you're doing is adding bacteria, you're not going to see this magical poop loop, as we say, of these magical nutrients being produced in the soil. And this is exactly what's happening. It's because the bacteria's job is to start working on the organic matter surface. They start working on the mineral components. When we look at these things under the microscope, you can see the bacteria, and they're literally covering stuff. They're covering the minerals. They produce alkaline glues, or is it acids? Alkaline, I guess,
Alkaline.
So they're producing alkaline glues that start to mineralize or break down or reduce those large molecules into smaller molecules. So in the context of sterile soil, so if you're doing things that are very antimicrobial, if you're adding a lot of pH up and down, if you're adding a lot of citric acid for pH adjusting, if you're spraying a lot of neem oil, if you're spraying Eagle 20, if you're spraying damn near any pesticide product that's antimicrobial, you are in this P, sterile soil to plant with barely bacteria situation. And in that situation, the bacteria are perfectly doing their job of tying up and immobilizing those nutrients to then be released by literally any predator to prey interaction.
Which is missing in that
It's completely missing.
environment. Yeah.
And so in the treatments with fungi and or bacterial feeding nematodes, there was more nitrogen being produced than
And
the
the plant
plants.
could even take up.
So now you're
Yeah.
having this regenerative thing. So everybody says regenerative. This is how you get to regenerative. You're not actually regenerating in a sterile or a just packaged bacteria product.
Yeah. Okay. In the amended study, the bacteria and fungi and the fungal feeding nematodes and the plant bacteria, fungi, and fungal feeding nematode treatments consistently immobilized more phosphorus than the bacteria, fungi, and the plant bacteria fungal treatments. The increased bacterial populations in the treatment with nematodes most likely immobilized much of the phosphorus, with a small portion also being immobilized into nematode biomass. Similarly, the plant bacteria and bacterial feeding nematode treatment immobilized significantly more phosphorus than the plant bacteria treatment. While most of the additional immobilization was due to the more rapidly growing plants in the plant bacteria and bacterial feeding nematode treatment, some of the phosphorus was also taken up by the larger bacterial population in the rhizosphere of the plant bacteria bacterial feeding nematode treatment. More phosphorus was immobilized in the plant bacteria and bacterial feeding nematode, plant bacteria fungi, and plant bacteria fungi and fungal feeding nematode treatments than in the plant or plant plus bacteria treatments because of the larger plant biomass attained in treatments with bacterial feeding nematodes or fungi.
So again, we're going to go back to this p-value. Whoops, hit the wrong button. Hold on. Can I go backwards? Ah, shit. Hold on. I blew it. Bear with me. Technical difficulties. There it is. So right here in this, this statement here in the amended study, so bacteria, fungi, fungal feeding nematodes, bacteria, fungi, fungal feeding nematode treatment consistently made more phosphorus stored immobile than any other treatments to an accuracy of less than 1% variation. That's insanely accurate, y'all. That's insanely consistent for dealing with bacteria and things of this nature. It's 2022.
Yeah. And they're attributing it to the bacteria as well.
Yep, exactly.
Yeah.
Mm-hmm. But nonetheless, extremely accurate science here. And again, phosphorus everywhere, phosphorus being held, phosphorus being produced, phosphorus being made available with simple interactions of organism to organisms. Some other interesting components of this study that we didn't get a chance to cover because there's just too much in it. But one of the things that really fascinated me was, in addition to all the things we've shared with you, they also took little scoops out of the root zone further and further away from the actual root hair. And then they charted and graphed those organism populations up against the root, on the root, and further away from the root. And I mean, they fell off a cliff, damn near. So that's another consideration for why we're interested in something like dichondra. If you're filling up that entire soil surface with literally any roots, you're now effectively using that substrate volume to produce, store, capture, and regenerate nutrients in your soil. So people that are just running no mulch layer, just your plant, you've got that big old pot. You get the benefit of stability in the root zone for water, so that's an advantage. But you're literally missing out on one of the most dramatic things of ecological succession, which is these predator-prey interactions, organisms eating other organisms, releasing these mineralized, then immobilized, then remineralized nutrients that are in extremely plant available and desirable forms for your plant. So yeah. You got anything to add while I answer this question over here? We got a question of what is the study?
Yeah, there it is.
The study is Interactions of bacteria, fungi, and their nematode grazers, effects on nutrient cycling and plant growth, written by Dr. Elaine Ingham here, and her husband, Russell Ingham, who's a nematologist. And the way you find that is you come over to Google Scholar, and you type in Ecological Monograph, and this gives all the results that are having to do with this title. The one we're looking for is this one. You click on it, and it'll take you to the website of the publication that it was in, which Ecological Monograph is a regular publication that is of the highest respect in science. You can read the abstract. Sometimes if you go to another website like JSTOR is one I've used and have a membership to. Just like Amazon, you go to look at a book, they'll give you a little bit of preview. Sometimes they give you more, but for the most part, they'll give you that first page. If you're enrolled in school or know somebody that's enrolled in a college or university, they can just go to the library and you can you can look it up just in the school library and typically download them for free. That's what I did. So, worst case, find a college kid if you're not one, or just even go to the community college and just say you're doing a research project and you need help looking something up. So we got some interesting comments here. Nature abhors a vacuum. Exactly.
Yeah.
Nothing is happening in a vacuum.
This is why we say diversity over singularity.
Yep.
Even 12 species from nature's standpoint is far too little. That's considered really singularity from nature's standpoint when she has, I think they're thinking there's like over 1 million different species of bacteria now is what Elaine was saying. So
yeah, I don't even know.
it's a lot of different species. I know there's over 5 million species of fungi. There's I know there's over 5,000 species of amoeba alone, so they all have their little niches and their jobs in nature and their preferred food sources. So, but the overall pattern is to get the food web in there because they do perform lots of functions and they stimulate the root biomass and the shoot biomass as well. So.
And control diseases and control pathogens and produce a tremendous amount of plant growth and water storage and actually sequestering carbon and all the things that we claim that regenerative agriculture is doing. You're not doing it with just bagged bacteria. Right. So we've come to about an hour, which is kind of the goal. If we got any last questions, drop them in. Go ahead and answer that question. There's about a 30-second delay. So
Smiley says, what do you recommend for a mulch layer? Something not contaminated.
How do you know? How do you know?
Preferably something not contaminated with arsenic or heavy metals or pesticides.
Which makes a narrow list, not gonna lie.
It can these days, to be honest, with all jokes aside, it truly can. So the best is, if you're just gonna do like a brown mulch, we'll say, and you're not talking about a green mulch, then if you just go to your local, like here we have, what's the name of that store?
I don't know. But we got
feed supply store
stores,
where you can get bales of stuff. They they tend to grow an off-season crop where they don't really care about it and they'll bale it up. Here we can get like a tri-bale or you can get straw or whatever. And those tend to be the best because they're not going to spend money on pesticides. They're not going to be.
They don't even fertilize it.
They don't even really fertilize it. So something like that is actually the best for a mulch. If you like the seed heads, then you can get something that has a seed head, and then you get both a green mulch and the brown mulch. But you might have to then chop and drop. So
tri-bale, and depending on where you're at in the country or even the state. In Southern California, they call it three-way bale.
Yeah.
Up here, they want to call it tri-grain or tri-bale. But essentially, it's typically 3 cool season crops like oats, rye, and barley or something along those lines. And it comes with the seed head. So it's a brown straw with the seed head. And that, in my opinion, is the best. You spread it out. You get the seed heads in there. You get a cover crop. You get a little bit of nitrogen. A lot of people are into grinding up barley and all that stuff. If you get just the natural seed head, it's less sugary. I think it's a little bit less volatile. And then you'll get a cover crop growing. Once it gets to about 8 to 10 inches, we then mow it. If you let it get too tall, it'll start to fall over and it'll actually cover the soil, and then it will actually start to reduce oxygen slightly. So you don't want that.
Yeah, that's a big thing with mulch, is you want to make sure that whatever you do, if you're not into growing grasses and you don't want to chop or anything like that, you just want to stick with a brown mulch, whatever you do, don't smother the soil.
Yeah.
Let it breathe, let it respire, let it, if you're getting fungus gnats, that's your biggest indicator to thin it out a little bit.
Yeah, exactly.
Yeah.
Yeah, but just slightly spread it out, somewhere between, if you think of a pizza, the coverage of pepperoni versus the coverage of a heavy cheese. You don't want to be all the way to heavy cheese, but you want to be more than pepperoni. So somewhere between how much pepperoni you put on and how much cheese you put on is you want to see a little bit of soil, at least in the beginning. You can always add more, but if you put a big fat layer, you actually slow down decomposition and it'll just sit there.
Yeah.
So you want to start lighter, let the system get going, just like starting a campfire at the beach, get some small kindling going, watch for that moisture to be absorbing in the straw layer, watch it to start decomposing, and then lightly sprinkle it on. It also helps if you kind of, let it do the pickup sticks thing. So spread it different directions, flock it out, let it interlace over and stack on itself, and, go from there.
All right.
So with that, I think we'll try to keep it tight around an hour. Thanks for everybody that came in, and
Thank you so much for joining us.
Exit music.
Hopefully we'll see you on Saturday.
Here we go. Thanks for
Bye.
coming.